and worked with Professor Lotarelli. Now he is currently taking care of the HLA typing for related and unrelated patients, research in immunogenetics, and in deregulatory cells and mixed fibers after transplantation. The title of this talk will be the role of different immunogenetic factors in the outcome of hematopoietic stem cell transplantation in thalassemia.
Thank you very much, Chairman. Thank you to Professor Deslani for inviting me here. Really a big pleasure to be back again in this beautiful country, in this beautiful city. I really enjoy, and I really feel like being back home when I come back here. And you know that we have a long-lasting cooperation with Professor Deslani in these years, and it's always been very, very nice to be involved in this kind of cooperation. So actually, I will my talk will be based on immunogenetics, and bone marrow transplantation. Actually, you know that bone marrow transplantation has. Let me start with this slide because it's just a little. These are excellent, but especially when I talk. So, bone marrow transplantation is a very now consolidated activity, and it's nice to observe, as you know, that in the very last year, maybe that we should focus a little bit more. Is it a focus? You know that it was started many years ago, and the experience of bone marrow transplantation, especially in thalassemia, was in the early 80s, and now many different, a lot of transplant has been done during the year. And but you all know that the two major adverse effects are graft-versus-host disease and the rejection. Of course, graft-versus-host disease are the donor cells that are going against the recipient, and the rejection is contrary. And you know that this has been mostly due to the fact that there are some allogeneic response, especially due to the major histocompatibility antigens. So the HLA may play a role that is crucial for this. And I would like to share with you this very nice picture in a review from John Hansen from the group of Seattle, in which he showed very clearly that the HLA genotypic match, the related transplant, definitely has a lower rate of graft-versus-host disease. And if you compare the related transplant with the completely matched, with no mismatch for the unrelated, you definitely get a better gap. So to make a transplant in a related setting is much better than in unrelated, the probability of not developing graft-versus-host disease. And of course, if you have a mismatch or two mismatches or three mismatches in an unrelated settings, the probability to increase the graft-versus-host disease is of course increased. But still, there is something that is not related to HLA, because even if in the better probability of finding a completely matched donor within the family, still graft-versus-host disease is a problem. So there must be something different, and of course many of the people that is involved in this kind of research has tried to define what could be the reason of this gap, of this why graft-versus-host disease or rejection still happens. So there could be other structures. And I'm going to talk to you today about the three important structures on the cells that are the minor histocompatibility antigens, the HLA-G, that is a non-classical HLA allele, and about the key receptor, the key semi-immunoglobulin-like receptors. And then we start with minor histocompatibility antigens, giving first an overview of what they are. Minor histocompatibility antigens are polymorphic peptides presented on the cell surface and are presented by the HLA alleles, antigens, but they are not HLA involved. They derive from allelic cellular proteins and are encoded by autosomal or Y chromosome genes, and they have a very important role in graft versus host disease, graft versus leukemia effect or rejection. And in general, they are biallelic and they are encoded in an immunogenic or non-immunogenic form. Of course, there has been many cytotoxic T cells that has been able to be developed against minor histocompatibility antigens, and the variation, there has been identified many of them all over the genome, but mostly of them are restricted to this number. So this is a list of the more important minor histocompatibility antigens that are these, and so these are the restriction of HLA. So this means that these antigens are presented in the context of these specific HLA alleles. So for example, let's take AJ1 that has a biallelic form, and one that is immunogenic, the other one that is not immunogenic. And this is due to the mutation in one of the positions. So a single mutation gives the possibility for this antigen to be immunogenic or non-immunogenic. And of course, there are many possibilities in which in the donor and the recipient settings, these two different immunogenic or non-immunogenic form can be presented. So if the donor and the recipient are matched for them, there will be no effect or no probability of effect. If there will be some differences, there could be some difference in the direction graft versus host or in the direction rejection. So, but let's just take a look, just for a second, to graft versus host disease, because of course minor histocompatibility antigens, like other structures, could be important. But just let's focus for a second, just a step back on what graft versus host disease is. We know that in order to declare that there is a graft versus host disease, an acute graft versus host disease, the graft must contain immunocompetent cells. The recipient must express tissue antigens but are not present in the transplant donor, and the recipient must be incapable of mounting an effective response to the transplanted cells. So this is a very complicated picture, but I would like to go inside with you in some details, and then go back to the minor histocompatibility antigens or to other structures. Because there are basically three different phases in graft versus host disease. The first one is the conditioning regimen. The insult that the patient receives with conditioning regimens definitely make a tissue damage that in some way involve the patient in a more effective way. It can be effective in a more strengthened way. Then there is a donor T cell activation in the second phase, in which you have what is called the storm of the T cells. And then there is a third part in which the inflammatory factors may occur. Let's take care only for a moment of these. When these cells of the donor and of the recipient come together and they in some way are able to play together, the donor lymphocytes recognize mismatches in HLA molecules or in minor histocompatibility antigens. The donor T cells proliferate, and of course, everything that happens here is influenced by the fact that there has been an inflammation due to the conditioning regimen. So you can have an antigen presentation that could be in a direct way if host cells directly with the APC present to the donor T cells the antigens, or indirect, because the donor cells may present the antigens that is derived from the recipient. And if we go back to the minor antigens, the minor antigens could be part of this system. And in fact, of course, if there is a tolerance in a setting in which the donor and the T cells share the same minor histocompatibility antigens. But if one of the minor histocompatibility antigens that we have seen before in the list is different, these may cause a self-recognition and activation, and in some way to provoke graft versus host disease. This is just a picture to show you that both class 1 molecules may present the antigens. That could be each antigens, but in this case could be a minor histocompatibility antigens, or also the class 2 molecules may do the same thing. And then, after this happens, we have a storm of cytokines in which many different cytokines are expressed, and they could in some way interact and produce damage to the tissues. And this is what happens in the third phase after the T donor or the T recipient has been activated. So let's see what in our experience, not only in our experience, lots of papers has been published recently about the influence of minor histocompatibility antigens in the outcome of the transplant. And each of them is taking care of the different minor histocompatibility antigens, and there are very different results. Actually, I must be, you must know that it's not clear yet how much the minor histocompatibility antigens can in some way influence the outcome of the transplant. Many different groups have different results. Nevertheless, we wanted to study in our setting of transplant what that are all related, and I will present to you only the related transplant that we have performed in Rome, the influence of these structures on the outcome of the transplant. We have studied in this setting 87 patients, all related, 17 with graft versus host disease, grade 2-4, 9 that rejected the transplant or didn't show any sign of failure. So there is a software in which you can insert your data relative to the minor histocompatibility antigen. You insert your results of the minor histocompatibility antigen. You insert the HLA typing of the donor and of the recipient, and the software gives you a kind of prediction if with the setting of that you are tied in the minor histocompatibility antigens in the context of that HLA specific typing, you have a probability of graft versus host disease or of rejection. So after that, the software will give you this prediction. If it's completely matched, you are supposed to have no event. If there is a vector in the versus graft-versus-host disease or rejection is something that we have to take care of. Now, in our setting of the patients, of 87 patients, 57 had a prediction of graft-versus-host disease with this software. And actually, 4 of them, 4 out of 57, really developed graft-versus-host disease grade 3, 4. Among the 50 patients with no graft-versus-host prediction, there actually none of them developed graft-versus-host disease. And this was a very important, significant statistical result. The similar results were obtained for rejection. Among 24 patients with a prediction of rejection with the kind of setting of minor histocompatibility antigens that we typed, 6 actually out of 24 had a rejection of a graft, and only 3 out of 63 with no prediction of rejection. And again, this was again a statistically significant result. So that in this analysis, we can conclude that minor histocompatibility antigens differences, especially in a related setting where HLA typing is compatible but not minor histocompatibility antigens are compatible, we can have a strong influence both on the rising of graft-versus-host disease and of rejection. There is a very nice study that has been carried by Stefan Spellman that has been published a few years ago about the minor histocompatibility antigens and the outcome of the transplant, in which he examined more than 600 patients. And again, he finds some correlation only for unrelated individuals. He finds a correlation only for rejection, in particular for CD51, that is a very important minor histocompatibility antigen. But this paper takes care of unrelated transplant, in which, there could be so many other structures that may account for graft-versus-host disease or rejection, and not for hemogglobinopathies. But still is a very important paper that I suggest you to follow. And we are going to publish our result very soon. Another structure that could be in some way involved in the outcome of the transplant and try to justify the high increase of graft-versus-host disease anyway, even if you are dealing with donor and the recipient that are HLA matched, is HLA-G. HLA-G is a part of a HLA, of course, system, but it's a non-classical molecule, and is very peculiar because it's not very polymorphic like the other classical gene. There are different forms that you can find both in the serum or in the surface of the cells. And there are 7 different HLA isoforms, and again, that could be bound to the surface of the cell or can be soluble. This molecule is very important because it is expressed on the maternal fetal, and it seems to protect. There are very many observations that suggest that the bound of HLA-G inhibit T and T cells and K T cells activity. So, for example, if we just look at this cartoon, it's very nice the way in which it works, because in a normal system in which T cells would recognize the alloantigens that could be just the differences in HLA structure antigens, these would evolve in a lytic activity, since, as we will see later, there are HLA on the surface, the NK would not kill the cells. In a system in which we would not have the HLA cell antigens presented, the T cells would not work, but this would be the case in which the NK eventually would kill the target cells. But in the presence of a very large amount of HLA-G antigens on the target cells, like trophoblasts, for example, in a maternal fetus system, the presence of HLA-G antigens inhibit the cytotoxic activity both of the T cells and of NK cells. This is a very important observation. This is why HLA-G is considered a heterogenic molecule. It's very nice how it works from a genetic point of view. At exon 5, there is an insertion that could in some way produce a deletion of the last part of the molecule. And this is a 14 base pair polymorphism that it could be present or not present. And if it's what happens when there is the insertion of these 14 base pair, we have an alternative splicing of the molecule that produces a short RNA of 92 base pair that is deleted, and this short RNA is more stable and produces more HLA-G. And producing more HLA-G, the hypothesis that you increase the immune tolerance. So that's why it's one of the important molecules to observe also in the setting of the transplant. So in this study that we have published very recently, we analyzed 147 patients transplanted in Rome or at the San Raffaele Hospital in Milan, with which we collaborate. The patients were classified as the patient classes, 1, 2, and 3, as you all know, based on the clinical, pre-transplant clinical conditions. They received the standard conditioning regimen. And we had 14 of them with the insertion of base 14 base pair, and 106 with a deletion. 58 of them had graft versus host disease, 18 rejected the transplant. So we wanted to observe if the presence of these base pair insertion was in some way correlated with a less increase of graft versus host disease. So probably the number of the patient is not very too much large, but this number is not completely statistical, but there is a very important trend showing that patients with the presence of insertion of 14 base pair has a much lower probability to develop graft versus host disease. This is not influencing the rejection or the thalassemia-free survival. And this confirms what has been also observed by Professor Lananza in a different study in an unrelated setting, in which again the presence of these 14 base pair segment is correlated to a better outcome relatively to the presence of graft versus host disease. Now, killer immunoglobulin cells, the killer receptor. This is a very complicated story. We all know that NK cells are very important in the adaptive immune response, that are the very first cells that comes out after the transplant has been done in terms of immune reconstitution, and are very important for the innate immune response. And they work with together in concern with T cells, of course. There are classically large granular lymphocytes. They express the CD56 and the CD16 on the surface of the cells. They do not express the CD3, that is characteristic of a T lymphocyte, as you all know. And they are specifically able to eliminate tumor cells as virus-infected cells, of course. They produce interferon gamma, and they are characterized by the ability to kill tumor cells that do not express HLA. So the lack of HLA antigens of the cells is very important in order to let them work. The kill proteins are receptors that are expressed on the cells of the NK cells. And they are very crucial. And there are many different here, as you can see on this cartoon. Some of these here has an inhibitory activity. Some of these cells has an activating activity. And we'll see in a while what I mean with these. But nevertheless, you have to remember that these kill receptors are expressed. And in the context of the bone marrow transplantation, of course, all these cells, biohistocompatibility antigens, HLA-G, kill receptor are very important. So, from a genetic point of view, I would like to point out that they are on the chromosome 19 on this region, and and they have a fairly large amount of polymorphism, not as much as the HLA, of course, but they have a very important polymorphism. We know 15 genes, 2 pseudogenes, and 3 framework genes. And they are subdivided into haplotypes. Haplotype A, that is characterized by the fact that almost all of the genes are inhibitory, and only one is an activator gene. And a B haplotype, in which there is the presence of the genes that produce these key receptors, that has a very important activator activity. And the haplotype B actually are many different, because there are polymorphism within the haplotypes due to the fact that the activating genes are differently contained within these cells. It's very nice also to observe that some of them have 2 domains outside, and they have a short tails, intracytoplasmatic, that is meaning activator, and long tails, that is inhibitor. And what is very important to remember, the message I would like you to take home relatively to the here, is that these receptors are receptors on the NK surface of the cells, and they specifically bind the HLA molecules, especially the C molecules of HLA. So, any specific of the molecule that we saw before has a specific ligand, and the ligand can be different based on the amino acids that are composed. So let's just take a look at the way in which the key receptor work. This is NK cells. This is a killer activating receptor. One of those that say, okay, my job is to kill cells that has nothing to do with my body. This is a killer inhibitory receptor. So the blue one that we have seen, this would be the red one. They have specific HLA ligand. So if they are bound, if the receptor is bound to his specific ligand, he would not kill. Because he recognizes the HLA antigen that is expressed on the cells. And so there is no attack. Please, everything is peaceful. But if the HLA ligand is missing, the kill that is the activating receptor will kill the cell. So this is the system in which the NK work naturally against everything that is different. So, if you, for example, has a leukemic cells that has lost the HLA antigens, the NK, through the kill, do not recognize the cell like normal, because there is no longer on the surface the HLA classical that you would recognize. It kills the cell. So they reduce the risk of relapse, of infection, of rejection, of graft-versus-host disease based on the cells that they are facing. So this is a very new and very new in terms of discovery, but a very important system in that we can take advantage and it help in our immunological defense. There are different ways to look at the activity of the kill based on the hematopoietic stem cell transplantation. The Perugian way, the Memphis way, and the Stanford way. I don't want to just a few words with words. The Perugian kill system model is helpful for the transplant from haploidentical settings. But we are not talking today about the haploidentical, so I would escape these. But I just want to tell you that in the Perugian model that has been very successful, as you know, this is a study from Professor Velardi from Perugia. Basically, the recognition of a HLA is able to predict if the NK of the donor would kill or not the patient. And so this is very important eventually in order to try to make a graft-versus-leukemia effect, you know. The Memphis ligand is the one that I showed to you. It is the missing cell. Again, if you miss the HLA antigen, the donor NK would kill the target. Or the last one is based by Stanford, is based on the fact that the kill genotype, as I showed you before, the two haplotypes, the two major haplotypes, A and B, have different contents of activator or inhibitory genes. So based on our experience, forgetting Perugia because I'm not talking about haploidentical, I'm just talking about the related, we studied 92 patients and 92 donors, all transplanted from an HLA sibling-related compatible donor. 52 developed graft-versus-host disease grade 2-4, 4 grade 3-4, 9 rejected the transplant. We typed the donor and the recipient with a commercial kit, and we analyzed all the possible antigens. Analyzing the results through the Memphis model, in which we analyzed any single kill of the donor respect to the single HLA ligand. When we analyzed them one by one, we didn't find any correlation, nor for rejection, nor for graft-versus-host disease. When we analyzed the two of them together, both against the C1 group, that is the 2DL2 and 2DL3, actually we didn't get a statistically significant result, but still the patient in which the ligand, that ligand was absent and instead it was present in the donor, all of them, many of them rejected the transplant. Even if it's not statistical, bound to the fact that few patients, 92 patients is still a very short, small group. But more interestingly, when we analyze the patients based on the haplotype, so on the full contents of the genes, we had the possibility to have the genotype AA, AB, or BB. And we call this AB or BB BX. So we had 11 patient donors that were matched for the full haplotype. We had 12 patients that were mismatched, patient AA, donor BX. 10 patient BX and donors AA. And we had 59 patients that were matched for BX. In the first group, we had 27% of graft-versus-host disease grade 2-4. In the second, 58. In the third, 78. And in the last one, 24. So it's very clear that in our hand, if you pull together the patient and donor that were matched for the KIR receptor at the genetic point from a genetic point of view, you had only 25.7% of graft-versus-host disease grade 2-4. If you pull the other, you had 63% of graft-versus-host disease, and this is very significant. And this is a unique opportunity to study this system, because when you do this kind of analysis in the unrelated trans settings, you have so many other structures, antigen structures that could influence. So we really think that the KIR receptor and the KIR system can have will play, and is playing actually an important role in the outcome of the transplant, and particularly in the increasing of graft-versus-host disease. And this is also true for the minor histocompatibility antigens, is true for the HLA gene. So what we are going to develop in the next future, it will be a genetic curve in which to put together all the immunogenetic factors that could in some way important and that could be relevant for the outcome of the transplant. So of course these are all the people that I have to thank both in Rome, starting from Professor Garelli, Professor Gazier, Sodani, all the other colleagues from the clinicians, from the people in Milan, we are collaborating too. And of course, the people of my laboratory in Tor Vergata University in Rome. Thank you very much.
Thank you, Dr. Marco and Daniel. And the discussion will be by the end of the session.
Our second speaker will be Dr. Santina Acuto, Gene Therapy Research Unit, Division of Hematology, Villa Sofia Cervello Hospital, Italy. She is going to give a talk on genetic therapy for beta thalassemia stage of R and challenges it faces in human trials.
I would like to thank the organizer and the president of the conference to invite me and to give me the opportunity to exchange with you some information about gene therapy and to share with you some of our results regarding this new option of cure for beta thalassemia patients. It applies to the patients that has no HLA matched-related donor and consists in harvesting hematopoietic stem cells from bone marrow or from apheresis after mobilization of the cytokine in immunoselective in vitro pre-stimulated and transduced with a vector that contains a recombinant beta or gamma gene, return to the patient after non-myeloablative conditioning. Factors that influencing the outcome of gene therapy trial are several, and today I will speak about vector design and ex vivo transduction condition. As a vector design to improve expression of beta globin gene, a researcher has spent more than 20 years to identify the minimal sequences requirement for having the globin expression optimal, which means being linearly restricted, stage-specific, elevated to be therapeutics, and sustained over time and independent from position in the chromosome. Because the target cell is a hematopoietic stem cell that produces all the blood cells, but the vector must be expressed only in the very mature red cells. So many lentiviral vectors were developed in the last years with most of the features in common. They have the minimal regulatory sequences from the loss control region, which confer high specific and regulated level of expression, and then they have the genes with the minimum of regulatory sequences again. Now, gene therapy is becoming a reality with one of these vectors entered in a clinical trial done in Paris by LaBouche and collaborators, and other vectors are really entering in upcoming clinical trial. We are collaborating with Memorial Sloan Kettering Cancer Center, Dr. Michel Sadelaine, to do a trial with this virus. The first patient that was treated in French, the treatment failed because the cells were had damage after manipulation, and so the patient had severe cytopenia, and he received back the backup frozen cells. He stayed thalasssemic, transfusion dependent. The second patient treated with this vector successfully became transfusion independent, and he began to synthesize hemoglobin, adult hemoglobin, from the vector gene, and 1/3 of the total hemoglobin that he has in the blood, it comes from the vector. But studying the insertion sites, come out that one partially dominant clones emerge, and the majority of hemoglobin, adult hemoglobin, was synthesized by this clone. The clone emerged because a vector was integrated within a gene that was overexpressed, so deregulated, only in erythroid cells, which means that probably the erythroid enhancer present in the vector upregulated this gene. So vector must be improved, of course, for safety, but also for efficiency. This vector, the TNS9 vector, was the first one to rescue anemia in the mouse model, of thalassemia. This is the thalassemic mice. And with this vector, 10 years ago, we were able to correct anemia and to prevent the secondary organ damage and to have enough sustained over time hybrid human mouse hemoglobin. We performed in our center in Palermo a preclinical study using a TNS9, I'm sorry, I missed here, a TNS 9.3 vector, which is a different version of TNS9, different because we introduced several mutations for safety. Doing preclinical study means we select hematopoietic stem cells from the patients, we characterize them, then we study the effect of cytokine treatment and other culture conditions on the potency of these hematopoietic stem cells and on the transduction efficiency. And then we put them in an erythroid liquid culture, which is a culture that in 2 weeks develops red cells from the hematopoietic stem cell. This is a classical HPLC analysis from untransduced beta-0 thalassemic cells from a beta-0 patient in which there is no protein, HBA protein, but after transduction, this is the protein that is made, the hemoglobin is made from the vector expression. And we are able to see also correction of ineffective erythropoiesis in vitro in this system, in the erythroid system, because thalassemic cells start to develop and there is an accumulation in vitro of polychromatophilic cells and there are not or very few orthochromatic mature cells, whether we treat the cells with the vector, with the therapeutic vector, the pattern at the end of the culture after 15 days is quite the normal pattern of development. So now a phase 1 clinical trial is starting in the United States, and we did mobilization study on our patients, on Sicilian patients, and they will be, I guess, the first who will receive this gene therapy. Now, vector designed to improve efficacy but also safety. As I told you, there are some side effects here with gene therapy. And because when we have a vector that integrates in the genome, the vector is subject to negative effect from the surrounding chromatin, especially if it integrates in heterochromatin. So the vector can be silenced, or the expression can be validated, or the extinction over time. And this diminishing the powering of gene therapy, but also you need many more copies in order to get the therapeutic effect, because for the positional effect. One potential solution to overcome positional effect is the introduction, the inclusion of DNA elements in the vector that are called chromatin insulators. The chromatin insulators are natural elements that function by isolate, by creating a barrier when flanking a functional domain and preserve the functional domain from the encroachment of heterochromatin. But also they have another important function, which is they block enhancer if they are cloned between enhancer and promoter. Both of these features are really good tools to be used in gene therapy, because by the blocking enhancer activity we can prevent the enhancer of activating genes that are close with the site of deletion, and with the barrier activity we will increase the efficacy of the gene, and so we will diminish the copy necessary for cure. We characterize now, we passed the 5 years to characterize a new insulator element from sea argent, from DNA, from sea argent, which is very small, and which displays blocking enhancer activity and boundary activity, both in the autologous system, in the sea argent, but also in the human cell, and also in the erythroid milieu, which is able to block the locus control region strong enhancer to talk with the globin promoters. And so we isolate, in this way, we protect the retroviral vector from chromosomal position. We publish already these results, in that the SNS5, the name of this new insulator, works in preventing extinction of the expression, as well as the chicken globin loss insulator, which is the well-characterized insulator since now. Then we use this chromatin insulator that we patent now to improve the expression of the recombinant TNS9.3 globin vector. And we found first that this insulator cloned in platinum position does not affect the titer, which is one aspect very important, whether the chicken insulator, as you can see, dropped down the titer dramatically. Secondly, we demonstrate that the insulator has a better performance in expression in cell line, erythroid cell line, but also we have the same result in vivo studies where we can get to more than twice of expression from the vector isolated with SNS5. And the expression is stable in 6 weeks, 14 weeks, 40 weeks. The variability of this expression diminishing with in the presence of the insulator, and also we have all the clones, the split clones, express the vector. So now the second important aspect is the ex vivo transduction condition that influence a lot the outcome of gene therapy. As I told you, in the French trial, one patient failed because there was no efficient genetic correction, and the other has this clonal dysfunction because for the insertion. So to have optimal condition of transduction means having an equilibrium between efficiency and genotoxicity. Vector, when going into the cell, are like money for people. If money are a lot of money in the hand of few people is toxic, but this situation is efficient. So, in order to calculate the real number of vector in the cell, we cannot compute in the vector copy number per cell, because this situation is one vector copy number per cell, but this is also one vector copy number per cell in the book. So we need to do to check each cell in each colony developed in the material cellulosa how many vectors it contains. And the factors that influence this trans-infection efficiency are the number of vectors, the MOI, but also the number of specific receptors for the virus in the cell. And this number is related to the cytokine treatment, but also the cell cycle status, because a lentivirus is able to transfect the non-dividing cell, but it prefers cells that are prepared to entering in G1 cycle. So for this reason, we begin to perform ex vivo amplification of cord blood cell, hematopoietic stem cell, and we try to transfect them and to see if we can increase the efficiency of transduction, as I told you before. So right now we have done 10 ex vivo expansion of cord blood hematopoietic stem cell with this profile. So at day 4, 5, 6, still we have 90% of the cells that are CD34 cell, but they amplify, the number grow between 5 to 10-fold, which is a really powerful tool. Then we characterize the CD34 subpopulation of the cell with two main markers. One, CD133, is a marker for very early hematopoietic cell, whether the other marker is for committed cells. And we found that cord blood ex vivo expansion showed main staminal finding in that the population that is CD133 plus cells grow after 4 days, 5 days of expansion. And this is true for all the samples analyzed till now. Again, the characterization of colonies developed in methylcellulose after at time 0, after 4 days and after 8 days of ex vivo expansion demonstrate that the more immature colonies, the colonies that are able to get granulocyte, monocyte, and erythrocyte, the CFU gem doesn't decrease at least at day 4, day 5, and then dramatically decrease. So we are performing also in vivo studies to demonstrate the potency of this system. But then finally, in the expanded cell, we try to do transduction and to see the distribution of the vector. So here you have two experiments with the classical pre-stimulation medium, and here we have 4 experiments where we transduce cell after expansion, 4 days of expansion. As you can see, first, using a lentivirus that carry marker gene, a GFP gene, we get almost double of the cells were fluorescent. And when we look at the mean fluorescent channel, which means the amount of GFP in each cell, we found that the mean fluorescent channel was less in these cells, which means probably we have less copy per cell. So when we checked each cell, each colony in methylcellulose, we discovered that around 1 copy per cell in this condition, most of the cells that are transduced with many vectors inside, whether 90% of these cells are transduced with the 1 vector inside. And so, from all of this study, we hope, we have a dream also in the future. First, we want to develop more safe vector for the current gene therapy protocols that are ongoing. But secondly, we hope that every patient, thalassemic patient that will be born in the future, we can probably use material that usually is discarded, the cord blood material, select the hematopoietic stem cell, expand them in vitro, correct with the virus, do extensive analysis in vivo, in vitro, etc., and so prepare the medicine for the future when he will be able to have the transplantation back. And this is my hope even for a G, because we have a lot of Thalassemia people. I would like to thank all of my group, but also people who are collaborating in New York for the mobilization studies and in the University of Palermo for insulator activity, but also for zebrafish in vivo system that we have developed, and Michel Sadelaine and Isabel Rivera Memorial Sloan Academy for developing the vector. And we are starting to mobilize patients in a multi-center pilot trial in Sicily, and this trial is still open till the end of this year, and we are invited, you who is interested in, to enroll patients so we can share the protocol, because also the idea that we have is we don't need to do everything in every place, because performing gene therapy is very difficult, very expensive, $100,000 for each vector, but also, GMP condition, etc., very hard to implement in every place. And that's why we are mobilized patients. We are sending, for example, in the United States, or we are doing some experiments in the laboratory to see how long they survive, how long they are transducible. In the future, we can plan to mobilize a patient in one place, for example, one place like India or wherever, and to do gene transfer in another place. So welcome to everyone who wants to collaborate to mobilize patients, and I will be able to share with you protocol, email, and information. Thank you.
Thank you, Dr. Santina and Tutu, for your very illustrative talk, and it's now open for discussion. I have a question for Dr. Marco Andriani. Yes. Dr. Marco, are you using the Tregs in treatment of graft versus host disease?
I'm sorry?
The use of Tregs, CD4 positive, CD25 positive T cells in treatment of GVHD. Are you have any experience in this part?
We are not using Treg like a therapeutic tool. Nevertheless, in my presentation that I will give tomorrow, you will see and you will understand that we are studying lots of T regulatory cells in patients that develop mixed chimerism. You know that mixed chimerism is the contemporary presence of donor and recipient cells. And I think that it's a very important news also for the people like Dr. Acuto that is taking care of gene therapy. Because in these patients, we found many T regulatory cells, specific for donor recipients, and T regulatory cells that are able to suppress the activity between the donor and the recipient. But we are not using these cells like clinical treatment.
Thank you very much.
Thank you very much, Dr. Andriani, for this very interesting lecture and concluding for very, very interesting lecture as well. I'd like to ask as regards the immunogenetic factors you presented. Is there any way to avoid the effect of these immunogenetic factors for the success of stem cell transplantation in patients receiving either haploidentical or identical transplantation?
Yeah, well, let's say that we are basically at the beginning of understanding what is going on. Because even if there are so many accumulating experiences, data, actually still we do not know exactly I think that you are very right in pointing out the haploidentical setting, because in the haploidentical setting, again, I didn't show you the results, but the key differences and the autoreactivity that some cells has against the recipients in haploidentical settings is very important. So let's say that many transplant centers, taking advantage of the experience of these immunogenetic factors, choose the donor inside the family, father or mother, based on the autoreactivity that the care setting may provide. And this is very important, I guess, because it will be an helpful hand for the clinicians.
My question for Dr. Akitu, you are saying that you are doing this gene therapy in cord blood cells, right? Yes? Yeah, you are inviting us if we can collaborate. Can you collaborate? If we have a pregnant mother now, and we know that the common fix is the thalassemia patients, can we have the cord blood of this baby who is going to be born and send it to you in order to do a gene therapy and again resume this in the baby when he's born?
This will be in the future. I'm sorry if I was not clear. Right now, we are beginning to perform the first clinical trial of gene therapy in an adult together with the Memorial Sloan Kettering. In the future, we plan to do what I showed you, and the data that I present to you in cord blood, they are really preliminary. We have to demonstrate first that we doesn't misregulated the genome expression after expansion. And actually there is a very nice place from Christopher Baum where they have a good result that if they expand cells, and then they do gene transfer in mice, they reconstitute more polyclonal hematopoiesis and in a shorter time with a less degree of genotoxicity. So we hope that cord blood expanded ex vivo, because as you know we need enough cells will be a powerful in the future. But right now we want to collaborate with you for mobilize patients with different cytokine or previxafor and see which kind of cell we can get and try to transduce them. And for the patient it will be very important to know if he's a good mobilizer, if the cells are transduced very well, and if he is a candidate for the gene therapy, the current gene therapy.
For Dr. Sidaren in Paris, who did gene therapy for 2 patients, I think, with thalassemia, and I think that they are still in a good condition, right? Is this different from that?
The first patient is thalassemic, again transfusion dependent, because gene therapy, the first patient. The second patient is transfusion free. Until now, he's doing very well. We know from rumors that the trial is starting again with other patients.
Make a comment. One of the important things, you were doing a very nice example with the distribution of money, of course. One of the things that I would like to point out, and I'm sure that you know about our experience in studying the patients with mixed chimerism, with persistent mixed chimerism that receive the matched transplant, we have shown that after many years from the transplant, some of these patients are living with a proportion of donor and grafted cells very low. Some of them have less than 25% of donor cells. Nevertheless, when we observe the origin of the cells that were erythrocytes in the periphery, almost 100% of the red blood cells were of donor origin. So we don't know what is going on. We are starting, and again, tomorrow morning I will present these data. But these are very important news for people like Dr. Akuto, who is taking care of the gene therapy, because as long as we know right now, no one is able to transfect all the cells. Nevertheless, if the origin is cord blood or bone marrow or peripheral stem cells. So the model, the knowledge that naturally in a setting of a later transplant, few donor cells are able to produce 100% of erythrocyte is very important, because this means that there will be a competition between the transfected. So the scenario in the future will be not in an allogeneic setting, but in an autologous setting in which the transfected cells from the patient will be competitive with the non-transfected cells of the patient. And we don't know who will win. Probably she has a more clear idea than me.
May I comment on the comment? First, it's true that you need low chimerism in order for thalassemia bone marrow transplantation, but remember, when you have one cell from the donor, that cell has all the sequences required for high expression. When we connect one cell with the vector, we never get 100% of expression. So we do need more cells to be transfected first. Secondly, I personally don't like to reconstitute bone marrow with few cells, because these bring to more genotoxicity potential. Because if you push 10 cells to recover the old bone marrow, they will do the job, but this recovery will be polyclonal, which means dangerous anyway, because tumors are, as you know better than me, I'm not a doctor, are a balance between doses. If you transduce many cells and you recover it with 1,000 cells, they are not pushed to, so you have a polyclonal reconstitution. And cord blood cells are transducible better than other cells. So, you know, all of this news, I think, will come in near future with good news for the thalassemia cure.
Okay. Any more questions?
Thank you for the two very impressive. I'd like to thank you very much for two very impressive talks, but Professor Amal almost asked my questions. I want to add a little bit of clarification, because you said you were speaking about the HLA-G, where there is a gene, where there is insertion or deletion. So could there come the role of gene therapy with a very little modification? It's not a whole gene therapy, but it's only to help in improving the outcome of the...
Yeah, well, if I understand correctly your question, actually HLA-G insertion or deletion has nothing to do with what she was showing. It's just the potentiality of increasing the tolerogenic environment. Because if there are more molecules of HLA gene, there is more probability to have a tolerant environment.
Yeah, that's what I meant. If you can use this environment, because you will only need it for a very short period, and that's the problem in gene therapy, because the maintenance of the outcome of the patient is very expensive and very tedious, but if you will only use such a procedure
Yeah.
I don't know what's
No, you're right, of course. We are working in an allogeneic system, and she will work in an autologous system with a modification. Of course, everyone that is working with allogeneic cells will try to improve, and as Amal was saying, in order to have better results, to avoid graft-versus-host disease, to avoid rejection. Nevertheless, and these are streets that are probably potential new address. Yeah, it's.
Thank you very much.
Thank you.